So today, first of all, welcome to all of you. And today we host as a speaker of this seminar, we hold Joan Falcó-Roget. Joan is at that moment is PhD student at the Sano in Team Computer Vision, which has another name, Brian and Mo, which is led by Dr. Alex Crimi. Unfortunately, Alex is now on his leave, so I will try to introduce Jean. Joan has completed a bachelor degree in fundamental physics from the University of Barcelona, and then he obtained a Master of Science degree in physics condensed matter. But what is important from the, let's say, from the point of view of today's seminar in biological system, and this time it was from Autonomous University of Madrid. So, well, he moved from Barcelona to Madrid to get his degrees. And then in Sano, his research around neuroimaging and methods to study all of these degenerative diseases in brain. And Roger Jean is author of about 7 interesting publications, and I think that in the today talk he will be talking about results he obtained up to now and about plans for his research aiming at obtaining a PhD degree. If you would like to ask a question during his talk, please put this question on questions and answers or on chat. And of course, as usual, there will be a discussion after Jean talk. So Jean, floor, screen is yours.
Yeah, thank you. Thank you, Mariam. Hopefully you can hear me well. And I was told that the screen is seen, so I will start. So please interrupt me if something goes wrong. But so yeah, I will start. So yeah, thank you, Mariam, for the introduction. And as he said, I was trained as a physicist, but I'm moving towards more biological systems and especially the brain. So this is the title of what I will talk about, but this should be so that the talk will be organized in this way. So we will have a short introduction, basic overview of one of the problems, or the problems as how we understand how physicists can join medicine, which is quite interesting. And this will be discussed in 2 examples, which is point 3 and 4. More specifically, or more in depth, point 3. And then I will end up, I will wrap up in the conclusions and some references if someone is interested. So as I was saying before, this talk is part of some, let's say, like a bigger project, which can have, or for the moment is based on this title. And I am under the supervision of Alessandro here at Sano, but he's also in AGH for now. And I would like to also deeply acknowledge, sorry, that went by, Alberto from the University of Messina, who has been helping a lot, and also some other collaborators, and also Nestor, especially, who was my mentor back in Madrid, and we still work together on very interesting projects, also mentioning Kasia, where we have some fun related to quantum technologies. So let's dive into it. As an initial motivation of me joining neuroscience, and especially this project, would be somehow to try to understand current computational treatment of brain lesions, however these regions may occur, And how does one study the very complex problem of causal assessment in medicine and in more general aspects, brain computation? Because this is a very complex problem and there are solutions. But so motivation point 2 is also how solvable or tractable are these problems, and more importantly to me, to what degree of precision, right? The way I was trained during my background, we used to simplify things in order to understand them. And perhaps I will talk a bit more in the end, but in the brain, this solution does seem to have a limited approach where complexity is so inherent that reducing this complexity leads to certain problems. So some of the hypotheses that I will be trying to answer related to this motivation is to model or to understand the explicit contribution of the magnetic resonance signal that is present inside the focal lesions and whether they are important or not to understand different disorders of different types. Now in 1.2, the second point, is related to the fact that when we have a system and there is a perturbation on the system, no matter the nature of this perturbation, this causes a change in the system. And this change can be assessed or tried to be understood within some causality framework, right? And I would like to pose that if we depart from purely information theory, we can still reconstruct meaningful relationships. And as a third hypothesis is that, well, while I acknowledge that these solutions are only approximate, we can still obtain satisfactory results, at least to a certain level of accuracy. And as an extra, because everyone who is in neuroscience always asks these questions, whether we need like models or we just need some other type of data-driven approaches in this type of problems. So as a general outline, methods, this is a highly interdisciplinary field that requires multiple resources, computational, neuroimaging, also from dynamical systems theory, chaos, and of course, machine learning, some graph theory and algorithm, information theory, as I also mentioned. And what is crucial and possibly the most difficult part of this is that you can just, or at least I have felt that you can't just dive into math for some reason and forget about the neuroanatomy or the actual biology that is going on in the brain, right? So this is super important, and this is actually hard, and this is why this makes it an interdisciplinary field with which to write collaborations. You can end up with very interesting hypotheses and a very good understanding of the brain from a higher level perspective, which is mostly neurological or biological. Okay, so I will just start with one of the use cases that we are interested or that I studied in this particular case, which is the brain tumors, right? So brain tumors are quite present and they can be divided in these categories, for example, and the most prominent, the most present is meningioma, followed by glioma, and glioblastoma is the most common type of tumors within glioma, right? The prognosis is quite bad for glioma, especially high-grade glioma. So it is definitely one problem to understand, but it's a very difficult data to acquire because it's a very, very sensitive pathology. So I'm sure that most of us are aware that there are different ways to measure brain tumor in magnetic resonance imaging. Importantly, of course, structural types of imaging where you can identify quite well all the structures, and we are aware of initiatives like BRATS where they try to use artificial intelligence to serve a specific purpose of detecting the tumor. But it is also an interesting field, although not super explored, I think, I could be wrong, of course, where they are trying to use diffusion MRI to image the microstructure of the tumor, right? So in a way, I'm guessing, trying to avoid biopsies or much more invasive techniques. So this is definitely interesting to have in mind. And the question that I will ask is, how about functional MRI? So there are some studies, I will review them here very briefly, but it's not the most used modality. And probably it's, well, there might be a number of reasons as to why it's the case. But for example, in this study, they delineate the lesion and they in a voxel-wise manner. So every voxel is like a pixel in 3D, and then they compute the Pearson correlation or another similarity metrics between voxels, and then you end up with some sort of brain connectivity. And for the control population, you simply compare each voxel across the whole population, and you can end up with a normative sort of distribution. And then you do the same for one patient, and you can end up with some anomaly map, right? In this case. But it is important that in this work, the lesion was totally masked. So the lesion was not contributing to this anomaly map. So this is one important thing to have in mind. And because this other study that interestingly came up the same year did not mask. So essentially they did a very, very similar approach, just that they computed the Z-score instead of just the distribution. But they did include the tumor. And as you can see here, perhaps this example is quite clear. There is the tumor here identified with what I am guessing is T2 image or maybe FLAIR. And this anomaly score is essentially detecting the tumor, right? So one of my starting hypothesis was that, okay, so the tumor seems to be functionally, seems to be playing a role at the functional level, right? So can we explore this? No? So that was the main question. No? So we are once again with our image here with the tumor. As you can see, the tumor itself is not super visible in the functional MRI, but the question I wanted to ask is, can we detect heterogeneity in the signal of the tumor, and can we characterize its relationship to the previous patterns of global disconnection in resting state networks? No? So can we somehow add the contribution of the lesion to those anomaly maps that were said before? So let's make, as I said before, I like to simplify things. So let's say that this is our tumor, and the way we operate, very simple, actually following ideas from this Silvestri in brain communications last year. I think that when I started, this was a preprint, or either that or I just put the year wrong. And we once again put this lesion that I just outlined here into, and we overlap it to our all control population. And then we can once again end up with a profile of alterations of the signal in the control population. And once again, we can map our patient in this distribution. This is not from us. We don't have this sample size, but we would be interested in looking the regions in the default mode network. As identified by the Gordon translation, what is known, which consists of 44 default mode network regions. For those of you who don't know, the default mode network is a set of regions that are not activated during task, or alternatively activated when we are doing nothing, and they have been related to a bunch of neurological disorders and cognitive disorders as well. For that, we of course need segmentations of the lesions, and the data set we used only had segmentation of the whole tumor. So I should definitely acknowledge Ricardo Budai and Rosemary, who I guess is listening now, that allowed us to identify this inner part. And by having the whole and the inner part, or the tumor nodule or necrotic area, we can identify or separate the edema. So hopefully I made a, and this is just to showcase that, yeah, in principle, we cannot assume that these two regions are homogeneous, but as a first simplification, as I show here, we are gonna suggest, or we are gonna analyze the whole tumor. And later I will come back to this. So hopefully I made myself a bit clear onto how are we gonna framing the problem, and now we will understand. The first thing that did surprise me is that for every patient, although here I'm just showing one example, the red line shows the functional signal only present as an average present in the lesion, while the gray areas, the gray lines, sorry, showcase the functional signals of the same areas in the control population. So kind of justifies the whole idea of looking into it, because there doesn't seem to be a lot of a a lot of differences, right? So we need to take a much more precise approach. And just as another example, so if we take that these are regions from default mode network, so we know that they are interesting when the subject is not doing anything. And this is from one example control and one region also from the default mode network. It's not easy to see any sort of differences based on the type of oscillations, right? So as a second step, as I mentioned before, we can compute the Pearson correlation of this signal against this signal and this signal against this signal, and we end up with a network, with a functional network, which is bounded between 1 and -1, but although you can see most of the numbers are positive, for the subject, and one network for the control population with its standard deviation parameters or mean, or as we want to characterize. As another biomarker, I should also mention that we are computing the complexity of the network, which is sometimes referred to as the theta richness of the network, and it essentially measures how different is the distribution of Pearson of correlations as compared to a homogeneous distribution. And for example, in this subject, the dynamical richness, the complexity of the network is more or less similar. But as you can see, these two networks, they definitely don't look the same. So why do the question that I'm interested, or that apparently I cannot take off this. So, okay. Question that I'm interested in is how can one assess the goodness of these oscillations that I have shown you? So whether they are pathological, they are normal, they showcase some kind of information. And for that, I will use the Fourier transform. So I'm going to go to the power spectrum, and let's see if we can characterize once again this signal. So this is the plot once more of just one example patient, and I'm going to guide you through it. Hopefully it will be clear. And in blue, we have the healthy population, and hopefully there are some small error bars here. So that showcase more or less that this is quite, let's say, normalized or type of measure for this population. And these histograms are just the power spectrum, while the lines are the cumulative power spectrum. This is the autocorrelation function, and then we can also compute some differences in power. So we have all these quite large number of features in this domain, and we would like to understand these features and how they relate to the tumor itself. So a small example, if we have two signals, I'm trying to guide you through my reasoning. If we have two signals that are dephased, for example, in this case, pi, if we just average them, we lose the signal completely, right? And one thing to be noticed is that even though the signals are clearly different, they are the same when looked at the power spectrum, the frequency domain, excuse me. What happens if we have a more complex type of scenario where we still have sinusoidal signals, but we have some dephasing, some differences in frequency, and the average, which is the black line here, shows kind of follows kind of the both oscillations at the same time. But what's most important is that they are clearly distinct. That you can distinguish them very clearly if we plot their power spectra. Of course, this is a simplified example, but if we look at the area between two cumulative distributions, we have something that is similar here. So what we did essentially is we reduced all this complexity into a single number, which is essentially how different these two cumulative power distributions are, telling us that if this is the case, for example, where a signal is above, so we have a positive, negative DAS score, which we name dynamics alteration score. That means that the signal inside the tumor moves at a slower rate, right? It has a slower velocity than the control population. And we can do this for every subject, and we want to precisely map whether this number, or let's call it hopefully biomarker, helps us understand the patterns of the altered default mode networks. So what we do is essentially compare by Pearson correlations between the networks, and then we correlate the networks between with alterations in the dynamics of those networks. And we can see that if we look at the absolute value, so we don't care if they are faster or slower, there's a significant correlation, although we can discuss if there is a stereoskedasticity, but there is a significant relationship, linear relationship, where it tells us that the higher the alteration, the lower the similarity, right? And if this is true for the absolute value, that means that if we display the not absolute value, we see the opposite patterns for positive and negative. So in a way, as I wrote here, the velocity of oscillations uniquely affects the rearrangement of the network. If we study the complexity, as I mentioned before, as measured by the dynamics alteration, we see a similar but different pattern, where slower oscillations end up with a smaller complexity of the network, while faster oscillations result in a faster complexity of the network. And if this is true, once again, if we plotted just the absolute value of these scores, we lose at least some correspondence, right? Because that means that we just having this sort of pattern here as opposed to the previous one. Then we also wanted to assess whether the location of the tumor affects this the dynamics of the network, right? So previous studies, the studies that I have shown before, but that I did not mention, were saying or concluded that the actual position of the tumor in the brain, so as measured in XYZ coordinates, it's not a good descriptor of how the signal itself is altered. So we wanted to assess once again, like, is this alteration in the dynamics related to the distance or overlap between the tumor and the network? And once again, so kind of confirming what was already shown, there is no significant relationship between the exact location of the tumor, and it's probably something more of where exactly is the tumor rather than its position. However, this is an interesting case. So if we imagine our simplified model of the brain, our default mode network, where this could be prefrontal areas, temporal lobes, and some parietal or precuneus, and here we have our tumor as I was showing before. If we compute the average distance and the alterations of each region in the default mode network, there seems to be an interesting case where closer regions have faster oscillations, and if we move away from them, then the oscillations are slower. So that led us to propose what I was actually interested in, which is what if we compute the power spectra of the network and we compute the power spectra of the lesion itself, the best descriptor to understand the alterations at the global level for the network is to just consider the alterations in the dynamics of inside the lesion. And the correlation is astonishing, even though the small sample size. And what this means, or at least how I interpret this, is that the properties of the signal inside the lesion are a very, very good explanatory effect of the alterations that we will eventually see at a distributed scale in the brain. If we accept that, then we can go inside now the lesion itself, and we can try to map these alterations with different tumor features. So very simple and exploratory analysis tell us that once again the alterations in the dynamics, the score that I was mentioning before, rather than the power alone, have much more. They can explain, or at least they are much more present than just power. And if we just cluster them, these alterations, we find two more or less consistent clusters. Now, I was happy, but it turns out that it's not so clear. And if we actually measure, or in a way divide our data set into different groups and compute the dynamics alteration score of each group, and one could think that, oh, actually this score differentiates between different types of tumor, whether it's meningioma or not, whether it's a small tumor or a large tumor as measured by the median of the data set, a bit of the location, the grade of the tumor. So high-grade tumors have generally like worse prognosis, much worse prognosis than low-grade, and whether the tumor intersects the ventricles. Intersects means that it's touching and pushing the ventricles rather than growing inside the ventricles themselves. Now, these graphs look very pretty. Pretty, like at least can look a bit convincing, but those differences were not significant. Nonetheless, we do highlight this one because periventricularity, so whether the tumor intersects the ventricle or not, it is an important factor as to how the prognosis of the survival rate, right? It's not, I can't say that I really understand or remember the why exactly, but it was an interesting case mentioned to me by Fabio Sambataro, who is also one of the co-authors here. And I just wanted to highlight that this is actually interesting and important, but it was not significant. So if we ask the same questions, but we only look at the signal of the necrotic areas, the results are the same. Of course, significances may or may not be altered, but they stay the same. And also for the edema. So that means that maybe we can simplify the functional signal in the tumor. Functional, that does not mean the tumor itself, as just an homogeneous region. Small comment about the data set. Data sets, as I mentioned, they are quite scarce in this scenario. And actually, I should thank one of the reviewers for like insistingly pointing to this. And we did some permutation and bootstrapping analysis to consider whether a small mismatch in the times of repetition had an effect, and the answer was no. And if we just drastically dropped 7 samples, which is quite compromised in our already small data set, the relationships remained, of course, losing significance for the p-value, because without 7 patients, the size of the data set really decreased. But more importantly, the highlight were the alterations in the tumor or in the lesion and the alterations in the network. They remain very relatable. If we try to take a similar approach in using diffusion MRI, this becomes a bit more challenging because people have studied this a bit more than the previous case. So that means that you need to be much more careful about what you want to do. So very briefly, one possibility to study the contribution of the lesion or edema to the structural networks is by using what is called a free water algorithm, which essentially kind of considers that in the lesion you have tissue and then you have water, and the goal is to estimate the parameter of each let's call it compartment. This is called compartment, to kind of independently fit some signal for the lesion itself. However, it uses diffusion tensor imaging, which is not very precise when it comes to crossing fibers, and it is also a how is it called? Degenerate problem, which means that it has optimizing these parameters is highly complicated because there are lots of parameters, and it turns out that the initialization of those parameters critically affect the solution that is obtained. The other family of tractography algorithms, which is based in spherical constraint, spherical deconvolution, moves the signal into a sphere, and by using a kernel, one tries to fit different orientations for the fibers. Now, I know that this is very high level, but unfortunately, I do not have time to go into details. So I will just briefly mention that adding the contribution of the lesion to this pipeline is very complicated because it's unclear to me, or perhaps it is unknown, or maybe it's research that could be done. One would need to properly propose or estimate a kernel function. However, there is a proposition that claims that instead of using multiple tissues, just using one, it is a less harsh algorithm than multi-shell because it keeps some of the anisotropy in the signal, which is proposed here and evaluated as well here. And we based our solution to this. Why did I say we based? Because our data was multi-shell, but if we just run multi-shell algorithms in our tumors, even if masking or without masking explicitly, the multi-shell algorithm, as shown before, does not reconstruct any signal in the lesion. However, if we use this algorithm, we only in the lesion, we can get some signal. The point is, okay, so why would I use one and why would I not use the other? Well, if we have multi-shell data, it's difficult to acquire and it's very precise. So it is a shame to just throw away multi-shell data when you have it. So what we did is, like, probably as most of you already thought, it's like we reconstructed with multi-shell data outside and reconstructed with single shell data inside, and then we merge. I will be a bit more detailed later. Another problem that we noticed is that when we want to do tractography and we only track in the white matter, if we segment, the peritumoral regions, so usually are classified as gray matter. So any fiber that could potentially go inside, so following this orientation, would be stopped by the constraint. So we needed to surpass these two issues, and the solution that we proposed was, very briefly, to consider the two signals separately. So we have our anatomical image where we can delineate the lesion perfectly, and we also have the diffusion image where we can also use the mask to only track with single shell or track outside with multi-shell. Right, so we end up with these two tracts, one where only tracks outside, achieving very, very good networks that don't consider the lesion, and smaller networks that are considering only fibers that start within the lesion. So we are only tracking starting here, and this leaves us with two connectivity matrices, one for the lesion and one that does not contain the lesion, and then we can join them with whatever approach to obtain a more supposedly robust network. This is a short description of how we control, because it's not clear that we can just do this and we just sample a number of streamlines inside the lesion that is correspondent with healthy populations. And in order to avoid oversampling between rows, we just take the maximum. So to study the connectivity between one region that was located here and one region that's located here, it could happen two options, right? We could have this blue streamline that just does the full path, or we could have a streamline that is cut in half. But if one region was big enough, we would kind of be oversampling if we kept both. So some results that I can show you, but evaluating tractography is hard. And if we consider the multi-shell pipeline, so without all the problems that I have said to you, as you can see quite interestingly, this fasciculus here, sorry, I don't know if I said the name correctly, gets kind of cut, and we can follow it if we keep the signal, and this important corticospinal tract, which is quite interesting, gets entirely cut by the lesion. And even though it does not penetrate, so the problem is at the peritumoral area, sort of like at the outsides. And the same we can also track, depending on the tumor, something inside. And what's also quite astonishing in terms of for surgery purposes, the number of streamlines that intersect the lesion by not considering it is way smaller. So that means that the effect of cutting or extracting the tumor without being careful can have quite important consequences for post-surgical scenarios. So small test also to assess whether it made sense or not. It's just, okay, brain networks are assumed to have some power law distribution. They can be scale-free. They tend to be scale-free. And what we said, okay, let's have small tumors, because it's easy to see that if the tumor is small or non-existent, these two pipelines are equivalent. And actually the difference in the distribution of connections significantly disappears when the tumor is small. And an also interesting fact is that differences between our pipeline, we call it hybrid, and the control, seem to be a bit larger, but not significantly different, right? So we are reconstructing more or less equivalent networks, but they are just more precise. Hopefully I have convinced you. small use case is that our data set had pre-surgery and post-surgery, and we have networks at the pre-surgery stage and post-surgery stage, and we just try to map them. These are some, I will go over this very quickly because it's not super important, but we put them in a machine learning framework, and we take some Bayesian approach to it using some prior, and then we kind of map and sample from the machine learning model to obtain some networks that, well, although the pipeline is very simple and the data set is very small, we can somehow control for overfitting. So the distribution of z-scores is normal, and we get quite accurate correspondence with the real ground truth. And actually what I was most interested in is what kind of inferences can I make. And the most important one, or the one that came significant, was that actually small tumors have easier patterns. The data set was balanced because I just split it in the 50th, in the second quartile, so small tumors and large tumors. So let's say that patterns of disruption after the surgery, they were more normative in the small case. So if someone, so these are two published works related to this, and if someone wants to scan them, I will get a glass of water right now, sorry, before diving into the next issue, which is more related to causality. And I will be brief because I am fascinated by that problem, but it's a very complex and complicated one because it involves a lot of fields, notwithstanding neuroscience, of course. So perhaps the question that tries to answer is what constitutes the basis of cognition and disorders of cognition. And while this is very, very general statement, it's clear that cognition or disorders, well, come from different sources, but they have to be related with computations that the brain is doing. However complex, maybe it emerges something from these computations. Yet, if we want to be more precise, we must depart from this naturalistic or philosophical point of view, and we need to embrace a paradigmatic approach. And medicine, well, and lesions present an interesting case where we have a clear distortion, which is the lesion in the case that I was showing before, the tumor, but we can also have different, less focal disorders. No. So there are traditionally these approaches. I'm not going to go over detail with that, or I'm not going to go into details, but the oldest, one of the interesting ones is structural and dynamical causal modeling, that try to include the structure of the system, the physical structure of the system, or the dynamical structure of the system, which may not be the same, or other more machine learning-based, information theory-based, as Granger-Wiener influence modeling, which are quite old, but still very used, and also some newer approaches as well, based on purely information theory. However, these two, they have been shown actually quite interestingly and recently that they are very equivalent for the case of Granger causality and in the case of maximum likelihoods for the other type of estimator. So in essence, number 2 and 3 are the same, which provides a nice interpretation. However, we can use different estimation procedures. By the way, I do recommend this seminar by Joshua Tenenbaum at Yale. A different approach, so I wanted to, because I was also wanted to highlight that only a mad person would consider replacing these works and starting from scratch. And since I do consider myself to be decently not mad, then I will try to build upon another proposal that has been much less explored, especially for brain networks, which is based on cross-convergent mappings, which is essentially you have a dynamical system, you just reconstruct your what is called delayed embedding of each one of the two variables, and then you try to find a mapping, so kind of geometrical mapping or positional mapping between X and Y, and it turns out that this is important empirically, they are not the same. So you can extract some directed relationships between these two. A further work automatizes this mapping via using reservoir computing networks, which we have studied very well also in other contexts in neuroscience, that very briefly is a dynamical system that behaves in time and the system itself, so has interesting properties. And what's most important for our case is that if we have a system we scatter x and y, which is quite random, if we input the whole vector to the reservoir kind of organizes the points in this high-dimensional space, which is the population as measured by principal components. So this is actually a quite organized way of representing the system. In one of our latest works, we assess what are the basic requisites to produce non-symmetric mappings using this method. And very briefly, once again, the idea is, so we reduce them in this course because this is quite complex to assess causality in this scenario, where we have essentially two things. We need to ensure a significant prediction of x from y with respect to some surrogate as measured by this p-value here. And we also need to assess statistical significant evidence for this score being different than this one. So essentially, this score being different than 0, either positive or negative, encapsulating the different relationships. Of course, when I said surrogate, we are using iterative amplitude adjusted and permuted also Fourier transform. So once again, we're back to Fourier transform to generate our surrogates. And very short, we end up with this pipeline where we just fed. So we have a totally automatized way to establish the relationships between two different regions, where we just put the input to the reservoir, then we map it to the target, and we can plot for different lags, so how shifted the time series are, and we can assess whether there is a statistical relationship from one direction to another. And we tested, of course, in different scenarios, but I didn't want to test it in good scenarios. I wanted to test this in real scenarios, which is the data is noisy, data is not very well sampled in times, so we corrupt. And even if we corrupt with a lot of noise a logistic system with different interactions, we can get some information. So this is a well-known system from Sugihara in Science in 2012 proposal of the method. So we can get good results. And if we also get to more challenging simulations based specifically for fMRI, where there is this system with these interactions, and we also compare with Granger causality, just to have an idea whether we are good or not, and we actually get good correspondences. And even though I don't have time, like I said, to dive into the discussion about physically instantiated causality or not, I would like to point to three different scenarios. So this node here causes this one and this one, which are labeled 2 and 5. Now, Granger would probably establish a relationship between these two, known as spurious causality, but our score tells us that there is no influence from 2 to 5 and no influence from 5 to 2, but rather a combined structure that doesn't tell you that comes from the same source, but it does tell you that 2 is not related, like plainly to 5, and the other way around. If we look at directed, then we get good results where we get essentially disentangle 1 to 5 and not 5 to 1, which would be the opposite. And also, if we look at indirect causality, so from this node to this, it is clear that node number 1 causes 3, but it's an indirect causality, and what we get is a lowered, dampened score, but still significant. And the idea is to build, because the structure of the system, the different degrees of structure, so the first neighbors is this matrix, but if we square the matrix, we can get the second neighbor interactions. And this is important because in our optimization framework, we are not constraining to the structure, but we want to infer different structural levels or at different levels with which the interactions take place. Some also very brief summary of the results. So essentially, we apply this into data set of 224 human participants, and the main interaction happens from posterior regions to anterior regions, and with the temporal lobe a bit segregated. And even though this is kind of summarized here, and the implications is essentially that, well, in terms of higher order processing, these are speculated to be the first regions that come into play. And if we put into the context of existing works, we kind of get similar interactions where the dominant, seems to be the dominant regions are located in the back of the brain. We divided a priori just between temporal, anterior, and posterior regions, but if we just apply community detection algorithms to our networks, we essentially get a very good correspondence between the communities found and the structure and the anatomy of the brain. Once again, I need to emphasize that we are not constraining field structure. I just put here as a possibility because to estimate transfer entropy, we also need to use the delayed embeddings of our system to compute the transfer entropy estimate. And we also need to use the delayed embedding, so we can see x of t, xt minus tau in general, to reconstruct our cross-convergent mapping, or convergent cross-mapping, excuse me. So they have not been related, but of course, this is pure speculation, but given that we are using the same type of data, there seems to be, or there could be a relationship that could be studied from a theoretical point of view. So some works, once again, a preliminary study was also accepted at COSIGN and published as abstract, which is the Computational Systems Neuroscience meeting, which is the premier meeting of computational neuroscience. And also we have some work at bioRxiv that we want to submit soon. And lately, this was a very nice team effort actually last week that what was quite devastating for me mentally speaking. But we managed to, with Jamal, Wojciech, and Alex, to push this thing to a conference, which I like, given that it's double blind, so I will just keep us XXX, because I don't know if, I guess no one is watching, but maybe, who knows. So to sum up, this is my answer to my questions right now. And yes, we can take an explicit account of the lesion to gain at least an understanding of the disorders happening at the network level. And I just put this here because we have also some work very interesting led by Jamal, just recently submitted to the Journal of Neuroscience, where we are also studying stroke with data that both of us pre-processed, but he did most of the analysis. And there is an established solution to the same problem for stroke, but that is applied at the temporal domain rather than at the frequency domain. And it could be interesting to see if similar arguments hold as the one that I mentioned at the beginning of the talk. This is kind of a boring conclusion, but it is, yes, we can use, given appropriate considerations, we can use and automatize methods from dynamical systems, which is purely data-driven. So information theory or Granger causality requires the model of an autoregressive. So it uses an autoregressive model, which is kind of a constraint. Yes, we can still work and we can still push personalized and more precise models for medicine that take into account more and more details. Otherwise, like probably half of the research done in magnetic resonance imaging would go away. And this is just a bit of, for me, because I also like to think in more abstract terms. But even if we simplify the brain in whatever normative or methodological way that we want, we can still achieve something meaningful that helps us understand both brain order and disorder. And I personally would be also interested in understanding disorder. Some of the works published and already, and I just highlighted the ones that are especially relevant for this. So I have thought this is in workshop from Mikai, presenting the prediction method that I just went over very quickly. This is a work about the reservoirs, some review about reservoir computing, which we also use. And this is also collaborations with Marcin, and Kasia is not here. She is definitely involved in some other project that we are preparing related to whether or not we can use quantum computing to optimize certain biomarkers, especially modularity in our case. And this is the main work that I presented at the beginning. This is related to stroke, and the submission that exhausted me, and the preprint that was published in 2023. But we will resubmit after some feedback and also some feedback from COSIGN. Also some oral presentations related to all these works. And just to mention that I was also, after this causality work, I was also invited for a peer review to review a work and to master thesis that I supervised. Last but not least, thank you to the funding sources and of course Sano for paying me and letting me eat good food here in Krakow. And even though I am not officially with them, I would also like to acknowledge AGH and Kasia, CypherNet for computations, and now a quantum computing grant, and also the University of Madrid for providing me access to journals behind paywalls. And with that, I will end, and hopefully I am on time. Thank you.
Joan, many thanks for your talk. You are definitely on time, and now we have time for discussion, for questions and comments to what you have presented.
I will put the camera.
Okay. We have already the first participants of discussion. Olaf, please go on.
Thank you for the interesting talk. As always, it seems to be that the cohorts are a little bit small to do serious claims. So you are working, if I understood correctly, currently on a particular glioblastoma cohort that you have with, if I understood correctly, about 20 cases. What is needed in order to do a serious claim here? That will be the first question. In terms of a cohort that really would be convincing if we think about regulatory demands for such a biomarker to be entered into the clinics.
Yes, I don't know if there's a second question.
Well, the second question I can do at once, but this is a technical question:
okay.
how you do the regularization of the reservoir computing. But yeah.
Yes. Okay. So the first question, I absolutely agree with you. I actually had a lot of concerns at the beginning, and I share with you. And I think that I said biomarker, but I forgot to say that this is not a biomarker, because there is also an interesting situation where resting state fMRI is not very reliable. So the replicability or the intra-class correlation is not very high. So I'm not sure whether any biomarker based on resting state functional connectivity can actually be claimed as such. So we did a lot of, thanks to the reviewers, we did a lot of analysis with different resamplings of the data sets, even a study of how to assess heterogeneity in the data collection as well. And in all these analysis, the main trends seem to hold. But of course, we cannot claim a lot. So I do not claim a lot. So this study got published in a very okay journal, because I think that it was technically correct, not because it was super enlightening in terms of something that we can. My only purpose was to study the contribution of the signal. And however, I should also mention that with this type of data sets, we are actually not so bad in terms of sample size. So some study, we have a specific paragraph on that, and sample sizes of 20, 25 is very not uncommon in this. So, but I agree with you. And the second question, if it was related to the reservoir.
Well, let's stay with the first question. So I think your cohort comes from a partner in Italy, correct?
Yes. Well, no, they are in Ghent. So the data set is from Ghent. Yeah.
Okay. So isn't there any, let's say, additional cohort you could get from, let's say, Polish or other European facilities that you could contact in that respect?
Yes.
So
sorry.
I know, for example, well, I don't know. In Germany, I think there are large cohorts collected at Bonn, for example. So I don't know about this in too much detail, but as it is within the tumor thing, so might be also interesting to talk to Marissa about it, because she may have connections in that direction. So and from the Human Brain Project, they probably had a lot of interest in brain tumors, so they may have some contact that might be interesting there.
So I agree. Thank you for this. I was not aware of this situation in Bonn. But I also should mention that I take this as a preliminary study and also as a training study. And I also we are working together with some colleagues in the NASK in Poland, and also with Tomasz from the University of Valladolid, whom I met last week at Neuronus. And there is an idea of pushing forward these ideas with the National Center of Oncology here in Poland and some surgeons there to try to gather a better data set. Because it's true that there are probably other data sets out there, but then you need to take care about harmonization pipelines. So that means that acquiring data in different sites, they cannot be trivially merged. So this is something we considered. And actually, there is a paragraph in the paper that explains this. And we also have it in another problem, that you need to have both populations in the two sites in order to combine them. So we could not do it here. I could not manage to find any data set that was possible within the reviewing time frame that was given to us. But I agree absolutely with you, and it's good to know that also in Germany there are these institutions. So thank you for this. I don't know if I should go into the second question now, or
yeah, well, I think that is brief. Yeah, how do you do regularization? Well, there is also, although a little bit diminished, but it's there's also some risk of overfitting in the reservoir computing. So normally one applies either some rich regression or something else to do the regularization.
Yes, so the usage is quite interesting. It's very different from the one, actually, if I go back, it's probably, because we're not using it in the most canonical way. Yeah, here. So usually you put the time series as a sequence, and you fit one time point at a time, and then you get these temporal evolutions of the reservoir, which if you have echo state properties, then they go to the same place or not, depending on your purpose. But in this case, we are interested not in doing this, but fitting the whole time series as a vector, column vector, and fit it at the same time. And we extract a prediction all together. So this is definitely not the canonical way. And the purpose is to get to the reservoir to split the points in different neighborhoods, which can they be mapped by, as you see, incremental ridge regression. That was exactly your question. So
okay.
It's not about the initial. Of course, what you need is some non-linear function in the reservoir that is able to somehow, by using a lot of neurons, to have some sort of gated behavior where you have just different neighborhoods somewhere. And this is a nice example where all of this gets organized into two main principal components. And yeah, of course, we split the time series into test, and I think it's 80% this example and 20%, and we just like as you pointed out, I forgot to mention this incremental ridge regression. Yes.
Okay. So what is the non accounted for variance besides the first two principal components? So how much do you explain with the first two?
I don't know this. It's a good question. It's on my list to see if we can.
Okay.
Oh my
then
God,
I'll
I'm so
ask
sorry,
that
I
later.
forgot.
Okay. No
My
problem.
guess is that with the second one, my guess is that you explained quite a lot, no? Because they are quite separated.
Yeah.
And also,
okay.
they are quite separated. The first one, but sorry, I don't have it here. Sorry.
Okay.
And of course,
No
it should be noted that in every this will change in every instance of your problem, right? So yeah.
Yeah.
Okay. So
Thank you for the question.
many thanks, Olaf, for your question and discussion. Well, what well, I do not. I see Wiesław coming with a question and probably some suggestions. Wiesław, well, you can. I think that you can speak now.
Oh, thank you, Mariam. Can you hear me?
Yes.
Yes.
Okay, thank you for this talk. It's a really great topic. I'd like to have a better understanding from a clinical point of view, because I believe for clinicians, the basic question is, what is the type of tumor? What is the grade of tumor? And of course, there are numerous types. Some sources even give 120 different types of tumors. And if we limit to MR, there are two techniques. Spectroscopy imaging, which basically plays the role of imaging biopsy, or PWI, perfusion imaging. So why don't you use these two basic techniques in your work?
So DWI, we use it, but we do not. We use it in a different context, right? To reconstruct the structural networks rather than to look into the, or to try to characterize the tumor itself. I had an introduction slide, but I acknowledge that is a technique, definitely, and I think that if we get this project ongoing, I think that I would like to dive deeper into the diffusion part rather than the functional part, because as I said, as Olaf also mentioned, it's not a great biomarker, functional MRI. So I think that the diffusion is more promising. I was not aware of the other one, so I apologize for that. I guess that the easy answer to this question is that, well, in the data set, there was not this modality. So I was limited to the modalities that I had available there. So, but I do acknowledge that this is not published in a clinical journal. No, it's more biological. So of course, clinical claims should, if made, I did not intend to overstate them. I think that from using this work to clinics, there is a lot of intermediate steps to be done yet. So every critique or every related to the clinical application of this work is totally justified, and I cannot do anything else than agree with it.
Okay, so let's say if you have fMRI and you limit to fMRI, what is the meaning of fMRI in brain tumor? Because of course, we have some understanding what fMRI tells you about normal tissues. It still doesn't show the brain activity. It just shows change of magnetizations due to oxygenation. What is the meaning of fMRI signal in brain tumor? Does it show brain function, or maybe it's related to a higher vascularization of tumors, and it shows something different? Can you elaborate on that?
Could you repeat the last part? Sorry.
The last part is fMRI doesn't show brain function. It shows the change in oxygenation, desoxygenation in a signal, which of course changes with brain normal activity. What is the meaning of fMRI signal in brain tumor? Okay, because maybe it doesn't depict the brain function. Maybe it shows the fact that usually tumors have higher vascular networks, and fMRI signal, which depends on vascular flow, is changing.
This is the question, no? Like, what does it mean? It's unclear, but I think that one interpretation that we could do is that first, it's the properties of the signal in non-lesion tissues are relatable to the properties of the signal in lesioned tissues. So that's one of the things that I try to show, that what you have in a non-lesioned tissue is somehow shares something in common with the properties inside the lesion. And I think that what's more promising, or that should deserve more research, is the fact that, well, for some tumors, like, you still have white matter signal. You still have fiber tracts that go in and around the tumor. And if those fiber tracts are there, well, maybe it's not such a huge leap to say that maybe there is still some function preserved in certain areas of the tumor. But I don't think that we have, it needs to be further understood, for example, with some tract density imaging, or with some dynamic functional connectivity and tract weighted imaging. Also, these modalities are interesting to be understood, where you try to have some sort of common a space where you fit both the functional signal and the tractography, where you can sort of guess how much function is located, or is weighted, or is carried by a certain white matter voxel. Of course, I can't have an answer. I don't have a successful or satisfactory answer to your question. But if you ask me also, I'm not sure I fully understand or know what the functional signal is in normal tissue. There is some metabolic consumption of oxygen, but from that to neural activity is a huge jump as well. So I think that I can't say what is it. All I can say is that there seems to be both diffusion and functional signal that could be mapped, and perhaps it carries some significance. I don't know if I kind of answered the
questions are more for you, not for me, because of course you are doing this exciting project, and I think it's good that you look also from a clinical perspective and from the point of view of what sort of signals you are getting and how you should interpret the results. In some other project, you show that you use T1 weighted images to delineate tumor. Why don't you use T1 weighted with contrast, which better delineates the tumor?
Good. So good question. We don't have it. Once again, we did not find it. We did not find a multimodal data set that contained everything. That's one of the reasons why because we want to acquire one. So you can delineate, and maybe I can once again go back in time. If we have a T1 image, and the T1 and the tumor is of a specific type, you can still get or delineate the necrotic area. Of course, I could do that, but I kindly asked Rosemary and Ricardo to do that for me, because they have more experience, and perhaps this is enough.
It might be not
so yeah, this one is better.
Delineate
so even in T1, yeah, of course, with contrast enhanced and flare and T2, you get everything, like the enhancing part, the necrotic areas, and the edema, clearly, no? So they are very identifiable. But even in this simplified case where we don't have a multimodal data set, and once again, we really tried to find one, you can still kind of delineate some microstructure in the tumor. I should also mention that the reviewers asked the same questions as both of you. So you might, and we answered them in the same way. And it seems that we simply couldn't do more, right? So I guess that this needs to be understood as very preliminary work. And we had two clinicians also as co-authors as well. So and they also agreed, like, in terms of medical inferences or clinical inferences, it's very difficult to do it with this data set, right? So we just limited ourselves to pinpoint these factors, which is that when you have a tumor, you should not assume that the tumor is just there, not doing anything, both from diffusion and from fMRI.
Don't you think that your results and interpretation depends on the situation, whether the white matter tracts are penetrating the tumor or they are shifted by the tumor?
Absolutely. So infiltrating and non-infiltrating tumors, absolutely.
Do you distinguish them in your results, or you just use all tumors in the same way?
I'm going back to more this. No, so we try one. I went too far. So this is exactly trying to answer this question: whether we have a small tumor or a big tumor, the grade, low grade or high grade, whether the tumor intersects the ventricles or not, and whether the tumor is frontal, where we have more like. So yeah, we try to answer this, and it seems that we sort of find some differences, but they are not statistically significant. So I just put them because I wanted to showcase this green line here. But yeah, I mean, it's perhaps a biomarker, or sorry, not a biomarker. It's a number that needs to be further assessed with better sample sizes. So this is one of the aims that we would like to push this project with NASC and also with collaboration from Tomasz.
Okay.
Let's see if it's successful, of course.
And maybe the last question or comment. Some companies like BrainLab already provided some tools using DTI for tumor surgery. I think BrainLab is already on the market for about 2 decades. How different is your work with respect to the commercial state of the art?
Well, I think that it's unrealistic to think about this work to compete with existing commercial solutions.
I'm saying differentiation, not competing, of course.
Probably we are very behind, no? That's the main part. Like for example, we don't have a clear software pipeline, end-to-end pipeline where the client just opens the web page, opens the software, and gets a graphical user interface, and from the file that it's loaded, then it gets the information that the client wants. So we don't have that, and I think that it's more important. I would need to think what information would we showcase to the client, right? Because sometimes, as I have heard, the successful solution in industry is not always the best solution in terms of accuracy, or it's just a solution that solves certain practical challenges. And for that, I would need to ask surgeons, like first person, clinicians first person, whether this framework solves or can be incorporated into their daily pipelines, rather than just trying to push from my perspective or from my side a solution that I don't even know if someone will use.
Okay, so thank you so much and good luck.
Yes, well, many thanks for your comments and discussion. And well, I think that one of the important value which probably should be presented in a more, let's say, visible way is the method which was elaborated and which will be improved by Joan. So, well, the question is, it will be always question of data, and not only amount of data, number of data, but also, well, to which extent they are, let's say, how accurate this data are. But I think that what, in my understanding of this talk, what is the real value is a method which was elaborated up to now. So many thanks for your talk. And well, I think that we will somehow continue the discussion on this topic, as in a week from now, we will host as a speaker Dr. Philip Hart from the User Institute in Berlin, who will be talking about the similar subject. So I think that it will bring us a new vision on this important topic, or topic which is a leading in this Brian and Mola team at Sano. So thank you very much. And Philip, would you like to say? Yes, because Philip, I see that you raise your hand.
Yes, can you hear me?
Yes.
Yeah.
Okay. Yes, looking forward to my talk next week. Brief correction, I'm still working on my PhD.
But well, okay. But anyhow, we will host you. We'll host you in a week from now, and we will continue discussion on this, and probably it will be a kind of opportunity to some joint work with Joan. So many thanks to all of you. You are kindly invited to a seminar in a week from now. Thank you very much, and goodbye.
Thank you. Thank you. Bye. Looking forward to it. Bye bye.